Number 5. Higgs boson, the God particle. In 1964, a British theoretical physicist named Peter Higgs proposed an invisible field stretching across all of space, a field now known as the Higgs field that drags on particles as they move through it, giving them mass. Without it, electrons can't orbit atoms, atoms can't bond, matter can't form, and you and any living organism won't exist. Higgs wrote a paper, Two Physics Letters, describing the theoretical model, but it was rejected because they claimed it had no relevance to physics. After 25 years and over $10 billion, they built the Large Hadron Collider, a 17-mile ring accelerating protons to the speed of light. The press called it a doomsday device, thinking that smashing particles together might create mini black holes. The Higgs boson only appears once in every billion collisions. Finally, in July 2012, after smashing protons together trillions of times, they found it. And in 2013, Higgs was awarded the Nobel Prize in Physics. This discovery confirmed the rulebook that explains every force and every particle in existence. It's now the foundation of our understanding of mass within our universe. Number 4. Reprogramming cells. In 2005, every biologist agreed that cells only move forward, embryo to skin cell, embryo to neuron, one direction, permanent, and irreversible. That was developmental biology's foundation, and it had been taught as settled science for decades. Shinya Yamanaka, a former orthopedic surgeon turned researcher at Kyoto University, decided to test if it was actually true. In March 2006, he presented early results at an international conference in Canada. Researchers and colleagues said his approach was doomed to fail. 4 months later, he published a paper on induced pluripotent stem cells. He'd taken an ordinary mouse skin cell and injected 4 genes into it, and the cell rewound completely back to a blank embryonic state capable of becoming any tissue in the body. By 2007, he'd done it with human cells, and in 2012, he was awarded the Nobel Prize for the discovery that mature cells can be reprogrammed to pluripotent only 6 years after publication, making it one of the fastest recognitions in modern history. These reprogrammed cells are genetically identical to patients, meaning no immune rejection. Researchers now grow miniature human organs called organoids to test drugs before touching a single patient. A revolutionary discovery that exponentially benefits and accelerates every research, disease, drug, and potential pathogens. Number 3. Gene editing, rewriting biological code. Bacteria have been fighting viruses for 3 billion years. To survive, they evolved a system that clips out defeated virus DNA and stores it in their own genome as genetic memory. Next time that virus appears, the bacteria recognizes and destroys it. Scientists discovered the system in 1987 and named it CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. Most of the research community filed it under interesting but useless and moved on. That consensus lasted about 10 years. And in 2012, Jennifer Doudna and Emmanuel Charpentier cracked the mechanism. Their published paper in the Journal of Science showed the system could be reprogrammed with the potential to be used as a gene editing tool. Swap in a synthetic guide strand, and it cuts any DNA sequence you point it at in any organism. It probably sounds like an alien movie. And by 2020, it was adopted in labs around the world, and they were awarded the Nobel Prize in Chemistry for their discovery. Victoria Gray spent 34 years in crisis-level pain from sickle cell disease, a genetic disorder caused by a single error in a single gene. In 2019, she became the first patient treated with CRISPR. The FDA approved Casgevy in December 2023, the first CRISPR gene therapy in history. The most powerful biological editing tool in history came from studying bacteria's ancient immune memory. And this is only just the beginning. Number two, vaccine efficacy. Your body becomes the lab. For most of the 20th century, vaccine development took 10 to 15 years. Take a dead or weakened pathogen, inject it, and let the immune system learn. Then two scientists proposed skipping the pathogen entirely and just delivering the instructions. Katalin Karikó became obsessed with messenger RNA in the 1980s. Cells use it to carry genetic instructions. Inject synthetic messenger ribonucleic acid, and you could instruct cells to build their own medicine. Grant committees rejected her proposals. Her university demoted her. She had no lab, no funding, and no team. She kept working. In 2005, she and immunologist Drew Weissman submitted their breakthrough to Nature. It was rejected. They submitted to Science. Again, it was rejected. Reviewers called it not novel and they were not impressed. But then they sent it to Immunity and it got accepted. The discovery was that synthetic messenger ribonucleic acid triggered violent immune reactions because it contained a molecule called uridine. Natural human mRNAy uses a slightly modified version called pseudouridine. Swap one for the other, and the immune system stops attacking it. The mRNA could finally do its job. Weissman told Karikó their phones would ring, but nothing happened. It wasn't until 2020, with the COVID pandemic, the Pfizer-BioNTech vaccine went from genetic sequence to emergency authorization in under 12 months, with an unprecedented 95% efficacy. And in 2023, they were awarded the Nobel Prize. Their discovery turned a genetic sequence into a global lifeline, saving millions today and providing the foundation to save billions tomorrow. Number one, fusion ignition, the path towards Type 2 civilization. For 70 years, nuclear fusion was always 30 years away. Scientists understood the physics and the possibilities. Fuse hydrogen atoms together, the same process that powers every star in the universe, and you'll release enormous energy. No carbon emissions, no meltdown risk, and fuel pulled from seawater. Beautiful in theory. But the practicality of it sounds like a sci-fi movie. Imagine spending all your resources building a power plant that can power a city. Now imagine that power plant produces so much excess energy that it can use its own output to build two more power plants. Those two build four. The energy compounds from its own inception, and that is the promise of fusion. Not just clean energy, but a net positive loop where the output perpetually exceeds the input. It sounds like the kind of magic that violates the rules, and science has a long history of sounding apocalyptic before it sounds obvious. The National Ignition Facility in Livermore, California, had been attempting fusion since 2009. Hundreds of failed experiments and billions of dollars spent. Then in December 2022, the team fired 192 laser beams into a gold cylinder the size of a pencil eraser, containing a fuel capsule roughly half the size of a peppercorn. The lasers created conditions hotter than the core of the sun. The reaction produced 3.15 megajoules of energy from 2.05 megajoules of input. More energy out than in. The breakthrough of fusion ignition, and the first time in human history it had been done in a controlled setting. They repeated it again and again. By early 2024, one experiment yielded 5.2 megajoules from 2.2. With this breakthrough, the Commonwealth Fusion Systems is targeting a commercial reactor by the early 2030s. Energy poverty has become a solvable problem, and we are well on our way to becoming a Type 2 civilization. Thanks for watching.
Like and subscribe to see more.